All right, good morning again. We just did the case study, which is a physician's point of view. Let's do a scientist's point of view, an immunologist's point of view, and a vaccine maker's point of view. So I have a paper here that actually just came out. It came out right as this class was beginning. And when I saw this, I'm like, I need to show this to the class. This is late-breaking stuff. It's in the lab, and it hasn't moved to vaccine development yet, but I sure hope they're working on it because it looks like a new strategy for making a better flu vaccine. The title is Coupling Antigens from Multiple Subtypes of Influenza Can Broaden Antibody and T-Cell Responses. So we're going to build on the normal flu result, the normal flu vaccine. And we're going to say how to make it better by coupling the antigens rather than presenting them in the current situation, which is separate antigens. Separate but mixed, and it seems to matter whether they're coupled together. If you do this, you can broaden both your antibody and your T-cell responses, which means that you can have more of an immune response to the antigen, to the pathogen. So remember that we had a few things to talk about, but it's nice actually that because we want to talk about multiple immunizations. Most immunizations, you need to get them not just once, but you want to get them later, because you will react to them better and in a more long-lasting fashion if you get them 2 or 3 times. Some of them you have to get them like every season, like the flu season. Part of that is not just because your immune system forgets the old strain of flu, but because the flu itself mutates and evolves so that you have a new flu basically to deal with, and you have to keep presenting that flu, the new flu, to the immune system. So multiple immunizations tend to give you better immunity, and it just depends on the pathogen for how many we mean by multiple. Now, for young children, there are leftover maternal antibodies that can interfere. And so if you're giving an immunization to a young child, then after a couple of years, if they're less than 1 year old, you'll want to give them a later one when their immune system is fully fired up and they aren't dealing with maternal antibodies anymore. There could be a concurrent infection, especially if you have a low level of a certain virus that interferes with the immunization. It's helpful to get it another time when you don't have that interference. And you know that affinity, maturation, and class switching, these are things that happen later in the immune response, and they don't always happen every time. They happen at a certain threshold. So if you want to make sure that you have a lot of time to do affinity maturation, don't just have one go at it. Try to give your immune system 2 rounds of affinity maturation, and then you'll be less likely to be unlucky and to have a non-matured antibody. Class switching is also one of those things that happens after the initial activation. And, if you just keep doing this, you can build up a persistent antibody presence. If you keep showing it to your immune system, you're saying, this is important. Keep working on it. And the biggest reason, though, is that flu, we recommend a shot every year because there are multiple strains and they evolve. They change. This is the schedule right now, and so you see that there are some vaccines. I deliberately made this kind of small because the details don't really matter that much to us, except that if you look in the middle, you see a big, long bunch of yellow. Yellow is where you have to have repeated for certain ages for all children, and notice that this starts about 6 months for influenza and a certain type of COVID vaccine. And you're saying annual vaccination, 1 or 2 doses, depending on the child and depending on the recommendations of that year. The yellow bars that go over mean that you have to keep getting it every year because it's evolving faster than our immune system can keep up with. And it's just beneficial to do that. So you notice that many of these, the other ones at the top, like for hepatitis B, you actually get 1 at birth. And then you have a second dose at 1 or 2 months and a third dose around 1 year old. And then there are some catch-up vaccinations, but you don't have a recommended 4th dose on this chart at least. And so this is all for 0 to 18. This is a pediatric schedule that's recommended, and of course each person is different. That's one important reason why we have medical professionals to help interpret this general advice for the specific situation. The general advice for the flu situation is you have too much evolution, and you need to keep giving the flu vaccine every year. Remember that you've already seen influenza virus, and there's one thing: the influenza virus attaches, and this attachment point is usually the most vulnerable point because it's outside the cell where antibodies can reach, and in fact antibodies can block this. If there's enough antibodies around, they can get in the way so the virus can never get inside the cell. If it never gets inside the cell, it never gets access to the ribosomes and the protein-making machinery. But I do want to point out, as usual, Detmer uses more general words than we use in this class. For number 2, he says the spike protein of the virus inserts into the receptor like a key in a lock. And the only thing about this, that would work for COVID, for the SARS-CoV-2 virus. For influenza, it doesn't technically have a spike protein. We call it by a different name. We call it hemagglutinin. Part of this is because we knew that it agglutinated the cell. We didn't know it looked like a spike, so we didn't call it by a structural name like spike. And of course, we've kept the old name. Even now we look at it though, and it does look like a spike. It does act like the COVID spike protein. But you got to use the right name for it if you're going to read the literature. The right name for it is hemagglutinin, or HA for short. So here's a picture of the hemagglutinin. When it binds to the host receptor in green, it's the blue, red, and orange and yellow protein at the bottom. The virus is at the bottom, the infected cell is at the top. And so what happens is the blue part binds the host receptor, and then the blue part falls away, and the yellow part, the orange part, and the red part, they make a little grappling hook. Like the blue part falls away, and the grappling hook causes it to attach to the other cell membrane, and that red part is what brings the two membranes together. So the actual membrane fusion is the core of the protein. This is a brilliant and devious plan, because antibodies to the unless they catch it right in the act of pulling the membranes together, the ordinary hemagglutinin protein is going to look all blue. Antibodies will form against it. They can bind it, they can block it, but they can also, the virus can also mutate it like crazy, both to bind different receptors, and also if an antibody is binding the blue part, well, it's easy for the virus to mutate the blue part. It's not the crucial part for it to get into the cell. Notice how the blue part hides the red, orange, yellow part, and the red, orange, yellow part is the essential part. So it's kind of like, and you'll notice that malaria, if you study malaria or other pathogens that we have problems with, they tend to have defenses like this. So when we're giving the flu vaccine, the part that mutates is the blue part. So we need to make new antibodies for the newly mutated blue parts each time. The part in the middle doesn't really mutate that much. It can't because it has a very specific function. The blue part's only job is to cover up the yellow, orange, red part. So that's why it's hard. But that's what we've got to do. We've got to give it, we've got to try to keep up, we've got to try to have a broad response so it's not as easy for it to mutate the blue part and avoid our own cells. Okay? So remember we talked about this before, and I even showed you these numbers before, but I want you to look at them a little more closely. These are different influenza strains. Notice that all of these have the designation A at the very beginning. These are influenza A. There's also an influenza B that is a major different group of influenza. Now look up and down the antigenic subtype and see if you see any patterns. Especially recently, you keep seeing H1N1 over and over again and H3N2 over and over again. You see a few different ones, but mostly you see the H1N1 and H3N2 are the ones that keep reinfecting us. They're the antigenic subtypes. They're more similar to each other than they are to the other groups. And for example, you may have heard of the current bird flu referred to as H5N1, which is a different subtype. That's why people are worried about it. Even with that, though, see that there has been an H5N1 virus that was found in Hong Kong, and that's actually related to the flu vaccine, to the bird flu that's going around. So even within that, but you can definitely see H1N1 and H3N2. If you had to choose two subtypes, you would choose those. clearly, because those are the kinds that give us the most trouble, most infections. So basically they take these two and they put them into the flu vaccine. They don't just have one antigen, they actually make two separate antigens and they put them both in because they're just proteins. So you can fit more proteins easily into the vaccine. They're just the HA protein that will be the right shape and provoke the right antibody. Now there's also influenza B, which is not shown on this slide. They also have two major subtypes, and so they put the two A's and the two B's, and those go into the flu vaccine, and that seems to give us enough coverage so that the vaccine is worth it. Not perfect, but enough. So it takes months to make the vaccine, so we always have to guess which exact H1N1 and which exact H3N2 do we put in. You notice that there are different types that fall into those subtypes. There are different specific ones. Do we put into it, we can look right here, do we put the California version or the Texas version? Well, actually, it mutates enough, and we can try to predict and make an educated guess for which 4 sequences to put in the vaccine. And then we hope. It takes months to make the vaccine. I hope that this can be actually sped up, but it's hard to change big systems. And so it still takes months to make the vaccine. So sometime months before they make it, they pick the sequences within those particular 4 subtypes. They make the proteins, they put them in the vaccine, they ship them out, and they get them to people. On top of this, with the current vaccine, there are some people that will respond great to the current 4, and there are some people that will respond less well. They'll be exposed to the same circulating strain of virus, but they'll react differently and the vaccine will have trained them differently. It's like they're reading the same book, but they get different things out of it. Something that we're very used to these days. So the question is, why? Why does person A respond to the vaccine with a different set of antibodies, fundamentally different? It works like really well for person A, and person B also makes antibodies, makes the same number of antibodies, but they're not as good. Why does that happen? One theory is that it's time that matters. This is called original antigenic sin, and it's a red flag to me because I think this is a bad metaphor. Original sin is not a good metaphor for immunology. And so that shows me that the people that actually raise red flags, and it turns out that even though a lot of immunologists talked about this, I've never liked the idea. So according to original antigenic sin, the original strain you see is the most important. The first type of flu you see, you make a bunch of antibodies to it, and then you keep doing that same thing. Maybe you tweak it when you see other strains, but you're always locked in the past. Like you'll respond best to this while the virus has moved on. You're still stuck on trying to defend against the first one that you saw, which was when your immune system was most severely activated. Because everyone has a different infection history, therefore the infection history in this theory is what causes people to respond differently. Like you have a fixation on the wrong strain while this other person who responds better happens to have a fixation on a different strain that was, and it's the one that you actually got infected with, which can be variously different. So it matters which the virus was that you saw first. In this version, I've never liked this for another reason, because I think the immune system generally works well. And I'm surprised why you would have, in a context where viruses are constantly evolving, why would you have an immune system that doesn't work well? Now, there's a lot of things in biology that don't work well. This isn't an airtight argument. It's more of a suggestion and an inclination on my part. But I am suspicious when you say, well, the reason why people have different responses is because our immune system just isn't good enough. It isn't smart enough to realize that viruses are going to mutate. Viruses have been mutating for as long as the immune system has been around. So I don't see why memory would be a bad thing. Why would remembering the first thing only limit and divert the immune response toward that first thing only when we know viruses have always been evolution prone, mutation prone? Another theory is that the old memory is actually good. You can have cross-reaction where you have an old strain that sees a new strain and it sees it's a little bits of things that it like builds on the parts of it that it's seen before, and it actually will boost the immune response if you have the ability to see both of the different strains. This is a positive role for memory. I just want you to see how these are actually two different and opposing theories for how memory in the immune system works, which is a huge issue for the immune system in general. So in COVID, I actually never learned about original antigenic sin because I didn't work in this area of immunology. During COVID, people kept bringing it up, like all the immunologists think that we're going to have original antigenic sin, therefore COVID's going to come back and it's going to be really bad because we've all been immunized against this first strain. It's going to mutate, it's going to come back, it's going to be really bad, and it's going to get worse and worse. Where my view was, well, shouldn't memory account for that? Shouldn't memory be evolved to deal with an evolving virus? So again, I didn't really have a science about it. But based on this, I looked for good sides of cross-reactivity. And I think I found some. I did that with a student project during COVID. And during the next unit, we'll talk about that exact project. Because I think we found good cross-reactivity. So the idea is cross-reactivity limiting? Are you limited in your memory to not cross-react? Or is it a good thing that your memory can cross-react and it can broaden your immune response? How do you broaden the immune response to deal with newly mutated viruses? And that's where we come to this paper. This paper actually suggests that OAS is overblown. So I knew I liked it as soon as I saw it. I'm biased, right? We're all biased, and part of the point is that most people are biased in their own immune systems. So, the paper suggests that what's important is not what you see first, your infection history. What is important is your genes. Your MHC genes that you have, those will skew your immune response more than past exposure. Those matter more. If that's true, we can't control what infection history was. Maybe we could account for it in some way, but it's really hard to tell what you have been infected with 20 years ago. But what you can control is what peptides bind MHCs. You can tell which peptides like to bind certain MHCs, and you can give MHCs peptides that will work better. That's actually easy. And so this theory has a direct impact, and they went through and they showed how to make a vaccine better based on this theory. This is all about biochemistry of binding, because if it's about MHC genes, it's about peptide binding. Which ones bind which peptides? And if your MHC molecules bind a skewed group of peptides, and that's the problem, give them a bigger group of peptides so that the group of peptides won't be as skewed, and therefore the response based on those peptides won't be as skewed. That's what the coupling antigens does. So let's get into the data. The first data is that they have a big cohort, well-funded research. And by the way, this is from Mark M. Davis, who is probably my favorite immunologist. He just keeps doing stuff. I'll talk about his work again when we talk about cross-reactivity. It was his work that first convinced me that cross-reactivity was important. So there's 402 donors, and they were tested using an old standard assay called the hemagglutination inhibition assay. This is how immunologists have always looked at HA, how the hemagglutinin works in the protein. And they wanted to see how the antibodies would block that viral entry. So basically it's how you tell how well your antibodies work. And what they do is on this is they found that there were three vaccine subtypes that they offered. So the change in response for each donor after they were vaccinated, they would actually test the donor for their response against the H1. There's 4 different things. Remember, there's 2 Bs, and there's the H1 and the H3. Those are the 2 As. So what they would do is they would actually take the vaccine antigen. This is in the lab. They take their cells and they test to see how they respond to the different vaccine antigens. And what's interesting here is not only is each person's overall response different, each person's response to the different types of antigens. That's why we put 4 antigens in, because different people will respond to different antigens differently. So the H1 is shown in red, and by the way, they're going to keep these colors all the way through. the H1 antigen, there are some people that go from a low response before vaccination to a high response after vaccination. Overall, you get more response. See how the numbers on the right are always higher than the numbers on the left. That means after vaccination, you get more response overall to this antigen. But there's a lot of variation. Each person is an individual line going from left to right. So you can see that some people start off at 2.0 on H1, and then they go up to 8.0 on H1. Great. But there's a lot of 2.0s that stay down at 2.0 for H1. For H3, there's also a diversity, and for the B antigens, there's also a diversity. Basically, you have a lot of lines going up and some going down, more going up than going down, and that shows you that the immunization was actually helpful. The people going down probably already had something and had some kind of memory to this before vaccination. So overall, vaccination is good. The numbers go up, the lines go up, but each line goes up in a different way, and some lines never go up for the particular strain of the virus. So they wanted to map out each donor, and they found out they could see 4 groups. There are some people who are the lucky ones, who are the high responders. No matter what subtype they get, they always respond to it well. There are ones that respond well to H1 type subtypes when you give to them, H3, and then B subtypes. They're biased to respond to that particular antigen. And again, this is why we give them 4 antigens, so basically they're covered in one of the ways. The thing is, these people with the bias, they have an incomplete response because they're only responding to like 1 or 2 of the antigens. They're not responding to all 4. That's why you give them all 4, so you get those people. Is there a way that we can get more people to respond to more antigens? And that's the theory about this. So why do they focus on one antigen? Is it because they saw that H1 antigen first, therefore they're biased towards it? Or is it because their genes mean that they present the peptides from the H1 better, and therefore their genes are what's determining their H1 bias? So the HAI assay by itself can show bias, so they do a lot of this stuff, and I'm not going to show you it all, but I do want to show you they did ELISA. ELISA is important, and they showed that the H1 biased people tend to respond better to H1 antigens than to H3 or B antigens. And so you see in all of these, you have the red line is higher under the H1 bias red group. The blue line is higher under the H3 bias. Basically, that means they thought they had this bias. They confirmed that each one had that bias with ELISA. So, where does the bias come from? How can we separate out history from genes? Well, the first thing you do is you look at people who have the same genes. Identical twins, and again, for a twin cohort, this is a pretty big number, to have 78 people that are in the twin cohort. These tend to be adults, average age of 38, and they're relatively sex balanced, so that means that you don't have to worry about skewing the results that way. And what they do is they would actually so they just looked at the subtype bias. They classified them according to the way that their immune system would respond to the different subtypes. And 77% of the twin pairs had the same subtype bias. This is the kind of thing that indicates a genetic cause. And in many other studies, that might be enough for the study. This is just where they start, though, because they're immunologists. They want to see the immune system being skewed, not just the overall organism being skewed. So you give the twin pairs, or cells from the twin pairs, different ancestral flus, and you see how their immune cells respond to them. So the thing is, you take each twin pair and you label one purple X and one orange Y, and then you compare them to see, within each one, you want to see if they're in the H1 bias group, how do they respond to the ancestral strain. So the old strain, that would be like the original strain before it had time to mutate. So what they did is they see that there are differences. You see that the orange line and the purple line are different. There's definitely, the orange line is definitely lower than the purple line in every case. Maybe it's a little closer for the B subtypes, but it's definitely different for the H1 and H3 subtypes. Because they are responding differently to the ancestral strain, Therefore, they have different memories. Some of them have been exposed to the ancestral strain and they respond to it. Some of them have not. The memories don't explain the bias. They have the same bias, the same skew, but they don't have the same memories. Their ancestral strain response is different. So, okay, that's suggestive. Again, that would be enough for other papers. For this paper, then they go to how can you get people who've had the same history? Well, once they're grown up, they've had different histories. They can't control that. 1-year-olds, you can pretty much count on they've had the same history. Maybe a few of them have accidentally been infected by flu and you never saw it because it was asymptomatic. That's unlikely. So they're probably having the same history where the only history they have is the vaccine that they've received. And so the question is, do the 1-year-olds have a subtype bias? These have all received the same vaccine, and yet you have different groups. You have an H1 bias where you have more response to the H1 antigen. You have a blue H3 bias for some and not for others. You have the green biases as well. And in fact, you even have 2 different kinds of green bias. So it looks like the B is a little more complicated, but the A tells a very clear story, and the B seems to go along with that as well. 73% of the children, about the same percentage as the twins, they had a subtype bias despite having no history of infection and only the vaccination. So they all saw the same vaccine. They didn't have a chance to see anything else. This suggests that the differences are in the genes. They all have different genes, not in the history. So what are the genes that are crucial? The genes that are crucial that will vary most, remember the MHC is a highly variant locus. It's the most variant set of genes. And it is involved exactly in the immune response. So you look at that. MHC class 2 molecules for presenting exogenous peptides, but remember these are also important for viruses because they are required for T helper response. So the MHC molecules are biased depending on which genes you have. Look at this right here. I just circled the B genes. You have this group of MHC that if you get copies of these genes, remember you have 3 slots and you can get different ones from mother and father. So you can have up to 6 different ones. And so if you get one of those ones that's colored red, that's circled in green, then you will bind peptides from the B virus. This is actually computational, and this is the kind of thing that I did with students during COVID. So this is just taking peptides and what we know about the molecule, the MHC, and saying which peptides will fit into that MHC better. The B subtype has more peptides that will fit into those red MHCs better. If you get one of those MHCs, you're more likely to present more B peptides, less likely to present the H1 and H3. So if you're given all 4, you'll be presenting and binding the B peptides only, and you won't be presenting many of the H1 or H3 peptides. This group, if you get MHC genes in this group, you present the H3s well. And if you get some in this group, you'll present the H1s well. This is what we can do with a computer, and you will be doing things like this with a computer to be able to say which peptides can bind better. And of course, the computer, it's all we can do. We don't have labs that can handle this cell culture type stuff, and we can't do human experimentation, but they can. And so what you want to do is you want to back this up. The computer says the MHC genes will bias the immune cells toward particular subtypes. Well, you want to see, does this show up at the cell level? So now it's time for cell biology. What you do is you take peripheral blood mononuclear cells. This is an important acronym, PBMCs. It's what you see whenever you take the immune cells. So you're basically saying immune blood cells, okay? It's not technically just white blood cells, but it's a lot of them, you know? So PBMCs, this includes T cells, B cells, all the cells that have mononuclear, and they will be in the blood. And you'll take those from each donor, and you see that they did this. This is a lot of work, so they're only doing this for about 11 to 16 donors in total. But all this work, you take each pool of PBMCs, and you can give them different pools of peptides. You just make all the H1 peptides, and you give them, or the H3, or the Bs. The OR is important. And then you measure T cell activation for that. So remember, these are CD4 T cells. They have the other things, but we're not going to talk about those. Those are markers for activation. Those are the things that tell you that the T cell has been activated if you see them. And we're just going to say what the result is. We're not going to study that cell biology too much. So CD4 positive T cells, they were tested for these activation markers, and those from the biased person responded best to those peptides. In other words, the T helpers themselves are biased toward particular MHC2 peptide combinations. The peptides from H1 cause the H1 biased people cells to respond, whereas the blue and green other groups did not respond. So that's the graph on the left. The graph in the middle shows that if you're H3 biased, your cells will respond to the H3 peptides. They will not respond as well to H1 or B. And then the green B bias, if you have cells that are B biased, they will respond better to the B peptides. This is a very nice and clean result. So the question is why? Why are the cells biased, and how can we control that? Well, remember, B cells are clonal. Each B cell has one BCR, but they don't have clonal MHCs. They have all the MHCs that you have. So you internalize, but each B cell has to be biased because it has a biased BCR that binds one antigen, endocytoses it, and uses that antigen to make peptides that it presents that causes everything else to happen. So what you do is you actually connect all the antigens. Rather than having 4 different flu strains floating around, 4 different flu antigens floating around, each B cell gets one, makes peptides, and then is skewed toward that particular type. What if we just covalently, like tightly bound the different types together? In this case, H1 red, H3 blue, and the B subtypes, which are green. Instead of having them floating around separately, join them together. Then look what happens. On the regular situation is on the left. Each B cell gets one and endocytoses one antigen, presents one antigen, activates one type of T cell. You get a potential skewing response. If you just happen to have MHC that are skewed, you'll have skewed T cells. But each B cell will have multiple MHCs, right? So why not attach all the antigens together? That way, when one is endocytosed, the others are. And so instead of having just little red peptides inside the B cell, you have red, green, and blue peptides inside. It's just bound the red, but it brings red, green, and blue in, and that means it can interact not only with the T helpers that respond to red, but the T helpers that respond to blue and the T helpers that respond to green. That means broader help, more arrows. There's a lot of arrows, but the whole point is fewer arrows on the left, lots of arrows on the right, and more antibodies on the right. You see all those antibodies? You're getting red, green, and blue antibodies because you have more T helper help. This is another way to talk about it. Just remember the endocytosis. And if you have all of the genes linked together, so you have a multicolored, instead of just red, green, or blue, you have a multicolored antigen, the B cell is going to just bind one of those, but it brings all the rest of them in. And the MHCs are going to have those peptides to sample from. That means the MHC, the B cell can bind a red antigen, but it might present a green one. It depends on the genes that you have. And so you're just giving your MHC more chances. You're broadening the peptides available, and therefore you broaden the T helper part, and that broadens the whole immune response. Here's a case where they showed this happening. They took their coupled antigen that has it all of them together, and actually the other antigen was fluorescent. The binding antigen was not fluorescent. So when you have it just binding, if it was just binding and only that was endocytosed, you wouldn't see the inside of the cell get fluorescent. But look right here, you see lots of little green dots inside the cell. That means that you actually have antigen endocytosis, and if you have endocytosis, all that matters is that the right MHC comes next to the right peptide. That means that you have forced all subtypes to be endocytosed. This is the key idea, so I'm going to really hammer on this, and I'm going to have a test question on it. So then they tested this in cells, and again, they're getting more biological here. We start with, here's where we're getting very specific and biological, so I'm not going to show you the results from these, but I'm just going to show you the strategy, which is that they tested it in mice. And so, as typical, but Mark Davis has actually gave a big talk where he's like, we need to move away from using mice so much because we've healed all these diseases in mice. It's like, but we haven't healed them in humans. Even Mark Davis, though, needs to use mouse work to show that some of these things will help. And so they showed that they could predict from the type of antigen that they gave and the way that they gave it. They saw that if you coupled the antigens together, it worked a lot better than if they were floating around by themselves. And they also did it, this is actually cool. So organoids might sound kind of weird, but honestly, I think it's more ethical to use a human organoid than it is to use a whole mouse. You know, I want to move away from mouse research myself, and I like the idea that we can show it in mice, but we can show it in human organoids, and maybe someday we can move away from mouse work entirely, so that we are, I think the ethical considerations of a human organ is very different from the ethical considerations of a mouse complete organism. A human complete organism is, of course, another level entirely. So, in fact, you might say that you're not talking about levels at all, you're talking about qualitative differences. But all that is to say, organoids are cool. And so you take human tonsil tissue, you make single cells out of it, and then you grow an organoid out of it. That's a lot of biology that we don't need to know for this class. But to those organoids, you give them the free-floating vaccine, and then you give them the coupled heterologous, which means multicolored vaccine, where you have all four types coupled together. And also has a nice control. You give it a coupled vaccine, but it's all one color. All one type, either H1, H3, or the 2B types. And they found out from these, the coupled heterologous HA works best. You see how you have that from the mouse on top and on the bottom with the tonsils. That is the situation that they're saying works best. And they did, so all this should be enough, but they kept going because we're worried about a new strain of H5N1, which is not in any of these flu vaccines, and also we haven't seen it before. Unless we happen to work on a poultry farm, you probably have not been exposed to H5N1. That's why we're so worried about it. Well, what about if this can broaden the immune response against different H1 and H3 subtypes? What about an H5 subtype we've never seen before? Makes sense, but they actually showed that it does. So they used the avian HA, which is shown in yellow here, and that's the H5 antigen, and they showed how if the seasonal if you give someone the seasonal vaccine, then they don't have much response, of course. If you just give them the H5, it actually doesn't give that great of a response. One of the reasons why we're worried about it, we haven't seen anything like it before, and so we need a broader response to it. But what it does, if this theory is right, we should be able to couple the H5 to the seasonal vaccine and get the best response for the coupled part. Here they show that just plain avian by itself as orange, and then they show the combined one where they take the seasonal and they combine it with the orange as purple. And look at the bottom, how the purple lines are higher. You're seeing activation against the bird flu antigen from the system that has the coupled, and it's a lot higher than even the orange avian one. For the one on the far right, you see a little bit of response to the avian one, and you also, the cocktail works a little well. But look how much higher the purple response is. That means you have B cells that are responding and making antibodies to the avian flu better because it is coupled to the seasonal flu, which is related to something they've seen before. So they can bring back memories, and the memories can cross-react with something new. Memory is good, genes are important, and we can use this to make a better vaccine. Remember that the whole point here is the broadened T cell help. Because the coupling the antigen causes the B cells to present more peptides, causes more T helpers to be activated because they see those peptides. This is such an elegant combination of so many concepts. I'm definitely going to have some fun writing test questions about that. So make sure you study. So my conclusion, they don't come out and say OAS is wrong, original antigenic sin. But of course, this feeds into my own biases as a scientist. I've always had a bad feeling about OAS, and this just makes me convinced that it's wrong. It probably will take more to convince the diehards, and there's probably some ways in which it might be right in some cases. But I think it's more important to look at genes than history. And MHC genes are the most important. Of course, I love this because I've been looking at MHC genes my entire scientific career. So, of course, I think they're important. But I think I have evidence here. Hopefully you're convinced. You don't have to be convinced. You just have to answer the test questions accurately. Okay. So if we cross-link the antigens, we get B cells that present all the antigens to T helpers. Therefore, we get more T helpers. B cells still are clonal. They still have one antibody that they make, but they present multiple peptides. And so does this include, returning to our very first topic, does this include some of the effects of repeated vaccinations? Does this somehow include the one vaccination? You're kind of doing a repeated vaccination at once by having the peptides be in and having more T helpers activated. Maybe repeated vaccinations do broaden your T helper response. And this is one way to sort of do this. Maybe we don't need as many repeated vaccinations if we can make the first one be a better vaccination in the first place. And the bottom line, MHC molecules matter, and that means that when we looked at MHC peptide binding on computers during COVID, because we couldn't even get out of our houses during COVID, we could still do science, and that science actually looks like it really matters. So look for that story next, but I think this is enough for me to test on, and I will see you on Monday for that.